All right, so we're talking more photosynthesis here. We're going to describe where photosynthesis takes place in eukaryotes, the light reactions, and the Calvin cycle, and discuss different versions of the Calvin cycle, including C4 and CAM, and then really kind of emphasize the importance of photosynthesis. So here I have a picture of Vegetable Man. It looks pretty creepy, but it looks pretty realistic. So why is photosynthesis important? Photosynthesis uses energy from sunlight to produce glucose and other organic molecules. All food for humans ultimately comes from photosynthesis, whether it's whether we're eating corn, wheat, rice, bread, whether we're eating meat or fish. Ultimately, that energy was captured through photosynthesis. This drives all the ecosystems that we know, all plants, all animals depend upon photosynthesis. Also, in the larger sense, photosynthesis produces the energy that was stored as fossil fuels. And so most of the energy that humans use comes from photosynthesis. The other really big thing about photosynthesis is that it it produces oxygen gas as a byproduct. This is about 20% of our atmosphere and is what makes cellular respiration possible. So it's not just the food we eat, our building materials like wood, clothing like cotton, it's also the air we breathe. Okay. And in fact, photosynthesis uses the products of respiration as substrates, as the starting point. The CO2 that we exhale, photosynthesis uses to make new glucose using the light from the sun. Okay. And for us, respiration uses the products of photosynthesis, glucose and oxygen. So this is really important. And these two pathways then are kind of the opposite of each other, and they kind of run in reverse. And the production of glucose from G3P in the Calvin cycle even uses glycolysis pathway run in reverse. Likewise, in the light reactions, the principal proteins involved in the movement of electrons, which is electron transport, and in ATP production in plants are similar to those in the mitochondria that produce energy in electron transport after respiration. So they're really, things are really, really close here. These two cycles work well together, and that's what drives our biosphere. Okay, last time I talked about the structure of the leaves and the chloroplasts. The leaves are designed to let air and carbon dioxide into the leaf and let CO2, or excuse me, carbon dioxide into the leaf and to let oxygen out. Okay, the pigments and the enzymes for photosynthesis are all located in the chlorophyll. The pigments are in the membranes in the photosystems. Those are located in those stacks of internal membranes called grana. The liquid portion of the chloroplast that surrounds the thylakoid membranes contains the enzymes necessary for the Calvin cycle, the dark reactions. Okay, so the chemical reaction is 6 carbon dioxide and water are combined using light energy to form glucose. Some water is left over, and then 6 oxygen molecules. Okay, this occurs in 2 sets of related reactions. The light-dependent reactions, or I'll abbreviate them LDR. This occurs in the thylakoids with the 2 photosystems, and then the Calvin cycle or dark reactions, which occur in the liquid portion surrounding the thylakoids. That's the stroma. And there the main enzyme is rubisco. Okay. Last time we talked about the light-dependent reactions using chlorophyll. Light energy, photons from blue and red light can be absorbed in a chlorophyll molecule. This excites electrons so that they can be moved through from one chlorophyll to another. Okay, the excited electron is used to split water molecules, which will release oxygen that combines to form oxygen gas. And then the hydrogen ions and electrons are that are liberated from the splitting of water. The light-dependent reactions use water and those electrons and photons to make ATP and NADH. Those are the products that are going to enter the dark reaction. And so here you see the electron transport chain. The water is split by photosystem 2. The electrons move along the transport chain, and the hydrogen ions are are kind of stashed inside this membrane. Okay, and then eventually things get to the ATP synthase, which allows the hydrogen ions to diffuse through them, and that allows the production of ATP from adenosine diphosphate and loose phosphates. And then the hydrogen ions are attached to this NAD plus to make NADPH. Okay, so those are the products. This is the overall chain of reactions. This is the thylakoid membrane. Okay, so uh, chlorophyll is in the membrane. It's exciting electrons. The energy is used to split water. And we then use the hydrogen ions and electrons to make ATP and this NADPH. These hydrogens on NADPH are going to be hydrogens that are installed onto glucose, and ATP will be used to help that process happen. Okay, so here we go. ATP, NADH, DPH are used in the Calvin cycles. So the light-dependent reactions harvest energy from the sun to produce these two molecules. Those molecules are released into the stroma where the Calvin cycle takes place. Okay, last time I introduced the photosynthesis in the Calvin cycle. I'm not going to go into too much detail in the Calvin cycle, but to say that there are different versions of it. The normal version is called C3 photosynthesis, but there is a C4 version and a CAM version of the Calvin cycle. And these are both found in hot, dry environments. They are less efficient in a cooler, moist environment, but in a hot, dry environment, they're more efficient and they particularly save water. So you see these in cactus, in tropical plants, in crops that grow well in hot conditions, particularly corn. You see it in corn. Okay, these variations increase the efficiency during high temperature. Okay, and so we're going to kind of go into that a little bit here. The uh, C3 plants, these plants are the normal way the chloroplast does photosynthesis, and the photosynthesis all occurs in the same cell. The CO2 enters the stroma through these stomata that are open, and when this happens, we also always lose some water. In hot, dry climates, a process called photorespiration occurs, and this makes C3 photosynthesis less efficient. This photorespiration means that at high temperatures, the enzyme that attaches carbon dioxide and hydrogen, rubisco, instead of trying to attach hydrogens to carbon dioxide, it tries to attach hydrogens to oxygen. So a lot of the energy that's trapped in the light reactions is wasted doing this. Okay, and this, because the pores are open when this happens, this causes water loss. Okay, and so it's a big problem at high temperatures. Okay, in the Calvin cycle, what happens is that this can be modified to reduce water loss. And so the cells where photosynthesis occurs are kind of they separate CO2 attachment from the normal thing. And so they use a different enzyme called PEP carboxylase, which has a higher affinity for CO2 than oxygen. And it it fixes CO2 in compounds that have 4 carbons. Okay, and then they transfer that to a second cell, and then C3 respiration goes on. This happens in corn and in sugarcane, plants that normally do very well in high temperatures. Okay, a slightly different version of this is CAM, and this happens in cacti and pineapples. So C4 plants, corn, sugar, sorghum, and other grasses, they fix carbon using this PEP carboxylase in adjacent cells. So they don't do it in the same cell that the light reaction is going on. Then they transfer materials to bundle sheath cells where the Calvin cycle occurs. Uh, then in those cells, in these bundle sheath cells, that C4 compound is allowed to kind of degenerate, producing CO2 and pyruvate. Then the normal enzyme that creates a high concentration of CO2, and then carbon fixation occurs by rubisco and the normal C3 pathway. Okay, so what's the advantage to this? Well, the C4 pathway overcomes the problem of photorespiration and water loss through stomata. It requires additional ATP compared to the normal carbon cycle, C3 carbon cycle, but it reduces water loss enough, and where it's hot and dry, this is advantageous. And so corn is one of the plants that does really well in hot, dry conditions. And when photorespiration is common, this is really, really efficient. Cam plants do something similar. They also separate things. This is particularly in desert succulent plants like cacti or pineapples, things like that. Here, water loss is so severe that the stomata never open during the day, and they only open at night to reduce water loss. Okay, this again uses that PEP carboxylase, but they fix all their carbon dioxide at night and then limit the amount of photosynthesis they do during the day to the photosynthesis that uses up that CO2. So they're greatly limiting the amount of photosynthesis, but it doesn't really matter because water loss is so severe that you need to keep the stomata closed. Most of these plants are going to have really waxy leaves. Okay, so both CAM and C4 initially fix carbon dioxide into that C4 compound, and they do it in a different cell than in the cell that will complete the Calvin cycle. In CAM, the C4 pathway happens at night, and then the C3 pathway happens during the day. Okay, I want to end here. We're just PEP carboxylase is the enzyme that's added to C4 and CAM photosynthesis, and it's just to save carbon dioxide and prevent water loss. But I want to end here talking about why photosynthesis is important. Photosynthesis uses energy from sunlight to produce glucose. It's the only way glucose is produced. Virtually all organisms depend on this. Photosynthesis produces all the oxygen gas in the atmosphere. Okay, the carbon cycle, the movement of carbon in and out of the atmosphere, controls a lot of the features of the atmosphere and a lot of features of climate. So this is really, really important. I'll stop there. On Monday, I'll go through some examples of photosynthesis, and we'll start talking about the exam. All right.